Hello, dear students. In Science and Technology Part 2, we are going to start a new chapter, Life Process in Living Organisms. This is Part 1. This chapter is Part 1, and this is Module 1 of this chapter. Let us have a small overview of what we are going to learn in this chapter. In this chapter, we are going to learn about living organisms and their life process, some nutrients and energy efficiency, how living organisms produce energy, and about cell division, an important life process. Before moving into the chapter, let us recollect some basics. Can you recall how are the foodstuffs and their nutrient contents useful for the body? The body needs a variety of the nutrients like proteins, carbohydrates, fat, vitamins and minerals from the food we eat to stay healthy and also productive. What is the importance of balanced diet for the body? Nutrition is vital for your body and all of its system to function properly. By having good nutrition, it helps you maintain a healthy weight, reduce body fat, provide your body with energy, promote good sleep, and generally make you feel better. Which different functions are performed by muscles in body? The main function of the muscular system is movement, but it also helps stabilize our joints, maintain our posture, and generate heat during activity. Movement of our body can be voluntary and controlled by the skeletal muscles, or it can be involuntary and controlled by smooth muscles. What is the importance of digestive juices in digestive system? Digestive juices contains biocatalysts, otherwise called as enzymes, which enhances the process of digestion. Which system is in action for removal of waste materials produced in human body? The excretory system. It is responsible for removing the waste produced in our body. Now, what is the role of circulatory system in energy production? We know circulatory system allows the transport of energy-rich substances like glucose, fatty acids and other nutrients and also oxygen in the blood to the body cell so that they can produce the energy to run those cells. How are the various processes occurring in human body controlled and in how many ways? Organ system in the body are responsible for controlling various processes in the human body. They are done by coordination between various organ system as well as hormones. Hormones are all otherwise called as chemical coordinators. Let us begin the chapter. First, let us see how the living organisms and their life processes are associated. We know that there are various organ systems that are continuously performing functions in the body. Take for example, the systems like respiratory system, nervous system, digestive system, circulatory system which circulates food and nutrients throughout the body as well as oxygen, and also endocrine system that releases hormone in time. They are continuously performing functions in the body. Different external and internal organs are performing their functions independently but through a complete coordination. The organs are in need of continuous source of energy for this purpose. Carbohydrates, fats, lipids are main sources of this energy and it is harvested by mitochondria present in the cell. Along with it, oxygen is also needed by the body. All these are circulated via circulatory system and coordinated by control system of the body. Each life process contribute in its own way in the process of energy production, and functioning of all these life processes also requires energy. Human and other animals consume fruits and vegetables. Plants are autotrophs, they prepare their own food. They utilize some of the food for themselves, whereas remaining is stored. It is stored in roots, stems, fruit, etc. We consume these various plant materials and obtain different nutrients. Carbohydrates are obtained from milk, fruits, jaggery, canned sugar, vegetables and cereals like wheat, maize, millet, etc. We get 4 kilocalorie energy per gram of carbohydrates. Let us see how the living organisms produce energy. In living organisms, respiration occurs at two levels, at body level and also at cellular level. Oxygen and carbon dioxide are exchanged between body and the surrounding in case of respiration at body level. In case of respiration at cellular level, foodstuffs that we consume are oxidized either with the help of oxygen or without the help of oxygen. We are going to learn how the food we consume is going to get converted into energy. Before that, can you tell how many atoms of carbon, hydrogen and oxygen are respectively present in a molecule of glucose? In a molecule of glucose, there are 6 atoms of carbon, 12 atoms of hydrogen and 6 atoms of oxygen. Which types of chemical bonds are present in between all these atoms? All these atoms are bound by covalent bonding. In terms of chemistry, what happens actually when we say that a molecule is oxidized? When we see that a molecule is oxidized, it loses an electron, or we can say it gains a positive charge. In other words, it loses energy. We are going to learn about cellular respiration in detail. Carbohydrates of the food are the main source of energy for any living thing. This energy is obtained in the form of ATP. What is ATP? ATP means adenosine triphosphate. If you look at the structure of adenosine triphosphate, this is made up of a nitrogenous base called adenine and a pentose sugar, that is a sugar with 5 carbon called ribose, together called adenosine, and it has 3 phosphate group, that is why it is called triphosphate. Together it is called as ATP. Then glucose is oxidized step by step in the cells. This is called as cellular respiration. It occurs by two methods, either by anaerobic respiration, that is when oxygen is involved, and by anaerobic respiration where oxygen is not involved. In anaerobic respiration, glucose is oxidized in 3 steps. We will see in detail how it occurs. The first process in cellular respiration is glycolysis. What is meant by glycolysis? The word glyco means glucose and lysis means breaking down. So we have to understand that glucose is going to get breakdown. This process occurs in cytoplasm. So one molecule of glucose is oxidized step by step. So as a result, we obtained two molecules of pyruvic acid, ATP, NADH2 and water are obtained. If you see, this is the structure of glucose. See, then the glucose will be broken down and finally we get pyruvic acid plus ATP, which is the energy, and then NADH2, this is an enzyme, then water molecule. So the pyruvic acid, it is converted to a compound called acetyl coenzyme A for further processing. So two molecules of NADH2 and carbon dioxide are also released during this process. So the pyruvic acid will be converted into acetyl coenzyme A and two molecules of NADH2 and also carbon dioxide. So we will see in detail how this occurs, all the steps involved in glycolysis. We will learn glycolysis in detail. Glycolysis is a sequence of 10 enzyme catalyzed reactions. It occurs in cytoplasm. The most common type of glycolysis is the Embden-Meyerhof of Parnas, otherwise called as EMP pathway, which was discovered by Gustav Embden, Otto Meyerhof and Jacob Karel Parnas. The glycolysis pathway can be separated into two phases. The first is the preparatory phase wherein the ATP is consumed by the pathway itself, and the second is the payoff phase wherein ATP is produced. Glycolysis is used by all the cells in the body for energy generation. The final product of glycolysis is pyruvate in case of aerobic settings, that is with involving oxygen, and lactate in anaerobic conditions where there is no involvement of oxygen. Before learning glycolysis, let us learn the structure of glucose first. We know that glucose molecule consists of 6 carbon atoms, 6 oxygen and 12 nitrogen atoms. You can see glucose exhibits in two structures, that is as a straight chain compound or as a structure. This structure is called a ring structure or cyclic structure. Both are stable structures. You can see the carbon atoms are numbered from 1 to 6, and also there are certain arrangement of atoms, that is sometimes OH on the right side, sometimes OH on the left side. This arrangement or orientation of atoms are very important as they play a vital role in the chemical reactions involved by glucose. Okay, now we are seeing the 3D model of glucose, both as a straight chain and also as a ring formula. Anyway, for glycolysis, we will be seeing glucose in this ring structure. Let us learn about ATP also. ATP is adenosine triphosphate, which is an energy-rich molecule, and energy is stored in the bonds by which phosphate groups are attached to each other. So these molecules are stored in cells as per need. Chemically, ATP is triphosphate molecule formed from adenose ribonucleoside. It contains a nitrogenous compound called adenine and a pentose sugar ribose and 3 phosphate groups. As per the need, energy is derived by breaking the phosphate bond of ATP. Hence, ATP is called as energy currency of the cell. If one phosphate group is removed, it is called ADP, that is adenosine diphosphate. If one more phosphate group is removed, it is called AMP, that is adenosine monophosphate. You can now see the structure of ATP. You can see the energy is obtained by breaking the phosphate form. So when the phosphate bond is broken, it releases energy. So ATP is considered to be the highest energy form, where ADP and AMP are lowest energy forms. Let us learn glycolysis in detail now. The first step in glycolysis is the preparatory phase, where ATP will be consumed. You can see the cyclic structure of glucose here. The first step in glycolysis is the conversion of this glucose into glucose 6-phosphate. The name 6-phosphate is given because the phosphate group from this ATP is attached to the 6th carbon of glucose. This step is catalyzed by the enzyme hexokinase. Every step in glycolysis has its own enzyme to catalyze. In this step, first step, ATP is converted into ADP because one phosphate group is transferred to glucose and makes it glucose 6-phosphate. The second step is the conversion of glucose 6-phosphate into fructose 6-phosphate. The difference between glucose and fructose is that they are isomers, meaning the molecular formula will be same, but the arrangement of atoms, if you see, the arrangement of atoms will be different. Hence they are called isomers. This conversion is catalyzed by the enzyme phosphoglucoisomerase. You can see the step involved or the conversion is much related to the enzyme name also. You will see it throughout the glycolysis process. Now, the third step in glycolysis is again the conversion of fructose 6-phosphate. Here also, one ATP is converted into ADP, meaning the phosphate group in the ATP is again transferred to the fructose, making it 1,6 biphosphate. From the number, we understand that One more phosphate group is attached to the first carbon of fructose. Earlier the phosphate group was in 6th only. Now it is there in 1st and 6th. So it is called as biphosphate. This step is catalyzed by the enzyme phosphofructokinase. The next step, that is 4th step, here you will see the splitting up of fructose 1,6 biphosphate. Children, this biphosphate can also be called as bisphosphate, both are same. So now the 6 carbon fructose is going to get split up into 2, that is 2 3 carbon molecules. The first molecule is called dihydroxyacetone phosphate and the second is called glyceraldehyde 3-phosphate. These two are isomers. So after some time what happens? This dihydroxyacetone phosphate will get converted into glyceraldehyde 3-phosphate itself. It will be catalyzed by the enzyme isomerase. So after the 5th step, if you see, we will be having 2 molecules of glyceraldehyde 3-phosphate. So by the end of this preparatory phase, we will be having a molecule which is a 3 carbon molecule. We have started with glucose, which is a 6 carbon, and we are ending up with 2 3 carbon molecules. That molecule is glyceraldehyde 3-phosphate. With this, the preparatory phase of glycolysis is ending. Now we will move on to the payoff phase. That is, it starts with the 6th step of glycolysis. You can see in payoff phase, there are 2 molecules of glyceraldehyde 3-phosphate we obtained by the end of the preparatory phase. We will see in this phase how this glyceraldehyde is going to end up as a pyruvate. So first what happens? The glyceraldehyde 3-phosphate is converted into 1,3 biphosphoglycerate. So like fructose, here also adding up of one more phosphate group occurs. Here the phosphate group, this 2pi means that there are 2 inorganic phosphate groups separately. Earlier we were adding the phosphate from ATP. Here from the inorganic phosphate itself, that is taken as a nutrient. So this gets added and converts this glyceraldehyde 3-phosphate into 1,3 biphosphoglycerate. So the number indicates the phosphate groups are there in the 1st and 3rd carbon. So here you will see one more conversion, that is a hydrogen atom is getting removed. So this process is called dehydrogenation, and adding a phosphate group is called phosphorylation. So this step, in this step you can see 2 processes, that is dehydrogenation and also phosphorylation. See the name of the enzyme, it is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. So this is the enzyme that catalyzes this step. The next step, that is the 7th step, is that the conversion of 1,3 biphosphoglycerate to 3-phosphoglycerate. So this is carried out by the enzyme phosphoglycerokinase. Children, here you have to see the inorganic phosphate that got added from here is now given to the ADP and getting it converted to ATP. So you would have wondering why to change the same compound again and again, because this step, this conversion, that is generation of ATP, is very important. For this purpose only the conversion occurs. That is, the inorganic phosphate is now getting transferred to the ADP, which will be converted to ATP. It is catalyzed by the enzyme phosphoglycerokinase. The 8th step is called phosphoglyceromutase. This is the enzyme that catalyzes the 8th step, where 3-phosphoglycerate is now getting converted to 2-phosphoglycerate. You see the transfer of phosphate from third carbon to the second carbon. It is changing its place. The next step is, this is the ninth step, where the removal of water molecule occurs. Now you will know why the phosphate is getting converted from the third carbon to the second carbon. If it is still there, the removal of water molecule is not possible. So now water molecule will get eliminated from the third carbon, which is catalyzed by the enzyme enolase. Here it is written 2H2O because we are having 2-phosphoglycerate molecule, which now after removal of water it is called phosphoenol pyruvate. So there is one more step ahead of getting pyruvate. So this is the final step of glycolysis, where phosphoenol pyruvate, again this one more phosphate group, we don't want it. So what we have to do, we have to remove it. That is, we are getting it added up to this ADP, where this ADP is converted to ATP. So there are generation of 2 ATP in glycolysis. This step is catalyzed by the enzyme pyruvate kinase, and finally we got our final product pyruvate. So what will happen to this pyruvate? This pyruvate will now move on to another process called TCA cycle or citric acid cycle for further processing. So by this, payoff phase out, preparatory phase ends. So we will see the total rate of glycolysis. By the end of both the phases, glycolysis yields 2 ATP molecules, 2 pyruvate molecules, 2 NADH and 2 water molecules. Let us now summarize what we have learned so far in this module. We have learned living organisms and their life process, how energy is produced in living organisms. We have learned detailed the first step of cellular respiration, that is glycolysis. The rest two process of cellular respiration we will continue in our next module. Thank you.